it's a huge honor to be able to introduce our speaker tonight, Dr. Carolyn Bertozzi. She is one of the very few, rarely truly gifted individuals who can think in multi-dimensions at the same time. So she can think like a molecule, she can think like a cell, like a whole organism, and like a disease. And so her brain is operating in many different directions, and she can make progress in all of them. Early on, she developed a whole field of chemistry that allows scientists to link molecules together in a way that doesn't affect anything around them. So they're very efficient at linking up, but they don't affect anything else. And they're so specific that you can actually have these links occur within cells without disrupting the function of cells. And with that discovery and the work she's done in that area, she won the Nobel Prize in 2022. Over those years, beyond that work, she's been doing tremendous work in thinking about how to deal with cancer and the challenges associated with cancer. She's going to probably talk about some of that tonight. There's a property of cells that they tend to be wrapped in sugar molecules, and she's figured out that that's a way that they communicate with each other. And we'll be hearing about that tonight. She did her BA at Harvard and her PhD at Berkeley and a postdoc at UCSF, and I bring up all three of those institutions because I worked at all of them. Unfortunately, she stepped off the rails and went to Stanford. I won't hold that against her too much. But she's won the MacArthur Fellowship. She's the member of the National Academy of Science and Medicine, as well as the American Academy of Arts and Sciences. She's a Howard Hughes investigator. The list of things that she's done would take up a half an hour, and I don't have that time. So I'm going to welcome her here to talk about some of the more recent things she's doing. Thank you. All right. Okay, I get this.
What an honor. Oh, there I am. Okay. What a fantastic honor to be included in this amazing celebration. Thank you so much for inviting me. I was on the scientific advisory board for the Biodesign Institute back in the very early days, and I'm going to say it was probably around 2004 when I first made the trip here and saw what was at that time just one and only one building, now known as Building A. And it really was quite a revolutionary concept. I think even today, the Biodesign Institute is considered a very forward-looking model for a new way of doing science. But you were doing it 20 years ago. So I think ASU and Michael Crow and George Post and all of the leadership here were quite prescient in how bringing together the life sciences, physical sciences, engineering, and medicine could create transformative discoveries and technologies. And I'll admit that I was so inspired by what I learned from those years as an SAB member with biodesign that when I went back at the time to Berkeley and then about 10 years ago moved to Stanford, I have tried to recreate some of these interfaces and intersectionality at my own institution. So thank you very much for teaching not just the students at ASU, but the faculty across the globe how to do science in the best possible way. So with that, I thought what I would do tonight is just tell you a little bit about the kind of science that my lab is interested in. And Josh, thank you for that very kind introduction. And as you mentioned, at heart, I am a chemist. But I also took an interest, starting back in the late 1980s, when I was still a PhD student, in the field of glycobiology. And that is the biology of sugar molecules. And when most of us hear the word sugar, we immediately think of dessert, right? Food, things that you eat. And certainly sugar molecules are very important as foods, and we all have a rather tortured relationship with them in that regard. But there's a different universe of glycobiology that has not to do with eating food. Instead, it has to do with how cells recognize each other and how they communicate with each other, which turns out to be very important in the immune system, and it also has some relevance to cancer. And so that's what I'm going to talk about today. Okay, so let me just start with how I learned about sugars. When I was an undergraduate, so this takes us back to the mid-1980s, I took a cell biology course. And in that course, we learned about the molecules of life and how they work together to create biological function. We learned about proteins. We learned about DNA and RNA. And we barely learned anything about sugars because the world really didn't understand what complex carbohydrates, as we would have called them back then, were doing in our body other than maybe they were in the foods that we ate. What we did know in the 1980s is that all of our cells are coated in sugars. There is a shell of sugar molecules around the outside of every cell. And I was taking notes in my cell biology class, and I kept, by the way, I kept all of my notes from college. How many people here kept their notes from college? Okay. And wasn't your handwriting much better then than it is now? Because that's certainly true for me. So I had these beautiful, well-written notes, and I looked through my notes, and what I had written there about sugars was that sugars coat the cell similar to a peanut M&M. And this is what you learned at Harvard University about sugars, and that was about it. And at the time, what my professor taught us was that, oh, these sugars on the surface of our cells, we don't really know what they're there for, but maybe it's just like a peanut M&M, where, it's like a protective shell. And maybe it's just that simple, and maybe it's just that boring, is what we were taught. And does everyone remember that commercial from the 1970s about the purpose of that candy coating on the M&M? Because it protects that candy so that it doesn't melt in your hand, but it does melt. That's right. Okay. Well, fast forward, now many decades, and it turns out we have a lot more insight into the actual biological functions of this cell surface sugar coating. And nowadays, when people are using metaphors, they wouldn't use the peanut M&M probably. Instead, they might use the surface of the planet Earth. So just as the Earth is covered in vegetation, and if you were flying, as in this video, across very low, close to the surface of the planet Earth in a helicopter, and you look down, you would see all of these different features that are made from this vegetation. You would see open fields of grass. You would see trees of different sizes in clusters, some of them moving and swaying in the wind. That is probably more like what those complex carbohydrates look like on the surface of our cell. And this little graphic, which is embedded here, this is a computer-generated simulation of how complex carbohydrates on the surface of our cell might be moving as a cell flows through circulation in your bloodstream, for example. Where this blue part here, that's the surface of our cell, and the surface is made of what we call a plasma membrane. It's sort of a layer of fat, almost like that chocolate layer in the peanut M&M. That's the membrane. And anchored to the membrane are all kinds of different molecules with these long chains extending. Those are polymers. They're complex carbohydrates that are made up of simple sugars. So we now understand that this collection of complex carbohydrates that decorates the surface of our cell, this is a molecular landscape that contains biological information. So those sugar molecules are not random. They have a lot of very intricate, complex structures. And different cell types have different types of structures or different patterns. So if you're looking for metaphors, you can use the landscaping metaphors of the shrubbery on the planet Earth. You might also think of these sugars as creating a 2D barcode, which contains information about the underlying cell. And another cell might come along with a scanner that scans that barcode and learns something about the state of the cell. So by the time the 1990s rolled around, I had finished my PhD training and my postdoctoral training, and I was ready to start my own independent lab as an assistant professor at that time in the chemistry department at UC Berkeley. And what I decided early on in my own independent career is that what my lab might do is try to understand what are the sugar patterns trying to tell us. Like, what's the language of these sugars? And in order to read that language, we thought we needed some new technologies that were based in chemistry. So that became the focus of my early career, was developing the chemistry technologies, which included that chemistry that the Nobel Committee recognized 2 years ago, and to apply those chemistries to understand the language of the cell surface sugars. So now in the modern day, we have a much better understanding of what these sugars are trying to tell us. And I'll give you one example that probably everyone here would have some familiarity with, which is your blood group or your blood type. So how many people know their blood type? Uh-oh. You should go learn your blood type if you don't know your blood type. Let me be the one to tell you, if you didn't know, your blood type is determined by sugars. So you have structures of these complex carbohydrates on your blood cells, and that's what defines you as having either blood type A or B or O or AB. So how many people here are blood type O? I am. Lots of people. So what all of us blood type O folks share in common is we have this particular complex carbohydrate structure on our cells. And each of these different colored molecules is a different simple sugar, and they get connected together in such a way that creates the blood type O. Now, how many people have blood type A? Not so many, but quite a few. All right, so what the blood type A people have going for them is you have an enzyme inside your cells that puts this red sugar onto this green sugar, connects them together to make this structure. That is blood type A. And then how many people are blood type B? This is usually the rarest, at least in an audience like this. You have a slightly different enzyme that puts another green sugar onto the green sugar to make the blood type B structure. And if you're blood type AB, the universal recipient, lucky you, that means you got one enzyme from your mother, one enzyme from your father. You have a 50-50 mixture of the A and the B structures. Now, this was a really important discovery because it explained why a person with the blood type A cannot give blood to a person with blood type O. The person with the blood type O would see that as a foreign structure and raise an immune reaction against it and destroy those blood cells. So in order to do a blood transfusion, which is arguably the most important medical procedure of all time, you have to make sure that the blood type of the donor is compatible with the blood type of the recipient, okay? And by the way, the blood type O folks like me, we're precious, right? Because we're universal donors. Why is that? Well, the reason is, if you have blood type A or B, at some point, your body experienced blood type O as a precursor. So blood type O does not look foreign to you, right? So you can take O, or if you're blood type A, you can take A, okay? So this is an example of a discovery from glycobiology that was really quite transformative in the field of medicine. And when I started reading about discoveries like these during my training years, I was so fascinated, I just decided that this was going to be the area of biology that I would dedicate my career to. Now, of all the different ways in which cell surface sugars contribute to biology, I think the way that most intrigued me had to do with their contribution to diseases such as cancer. And you can look through the literature and you'll find evidence that dates back to the early 1960s that when healthy cells go through a malignant transformation and become cancer cells, there are some pretty dramatic changes that take place on the cell surface sugars. This has been known for a long time, but until recently, we really didn't have an idea as to why this might happen. And so my lab has been working on this now for the better part of 2 decades. What we do know is that if you inventory the pattern of sugars on healthy cells, you often find that there's a dramatic change in the density of sugars that have a particular simple sugar building block motif, which is this pink sugar called sialic acid. Now, the name sialic acid might lead a person to believe that this is a dangerous, toxic acid-type substance, but it's not, okay? That's just a terminology that comes from chemistry. It is a sugar, and people who study glycobiology often use a symbolic lexicon to describe these structures, and in my world, the sialic acid is always a pink diamond. And if you look at this cartoon, you'll notice that the healthy cell has a collection of these complex carbohydrates that look like trees with branches, and the pink diamonds are at the tip, and that's where you find sialic acids. They're at the tip, like the little leaves on the tree or the star on the Christmas tree. And healthy cells have what I like to think of as a well-manicured garden of these sialic acid sugars. But in the setting of cancer, that well-manicured garden just starts to overgrow, and it becomes like a tropical jungle. And this has been known now for more than half a century, and it's observed in lots of different kinds of cancers. You'll see this in breast cancers, ovarian cancers, pancreatic cancers, prostate cancers, leukemias. Probably more cancers than not share this property of having an overproduction of sialic acid. But why? Well, it turns out, and this is a discovery that came out of my lab over the last 20 years or so, it has to do with the immune system. Now, many people here have probably heard of the term cancer immune therapy. Is that kind of a household phrase nowadays? And I imagine that at ASU, you probably have some leading researchers that are studying cancer immune therapy. It's a really important cutting-edge area in oncology. If you're not familiar with the relationship between cancer and the immune system, I will teach you about it right now. So first of all, immune cells, these are the white blood cells that course through your body, as opposed to the red blood cells, which are the erythrocytes. The immune cells are the white blood cells. We sometimes call them leukocytes. And their job is to travel around your body all day and all night and test each of your cells to determine whether they're healthy or damaged. And if an immune cell stumbles across a cell from your body that's damaged, that immune cell has the job of getting rid of it. Keeps your body nice and clean. Healthy cells get to stay. Damaged cells, they get kicked out. The way that your immune cells can determine whether a cell is healthy or damaged is to snuggle up against it and literally form what we call a synapse. So the two cells are in contact with each other. They're touching each other. It's almost as if that immune cell is tasting this target cell to determine good, like yummy, or bad, yucky, okay? Most of the time, this works really well for us because a cancer cell is a quintessential example of a damaged cell. Cancer cells have damage in their DNA, which causes damage in their biochemistry, which causes them to start becoming very antisocial, and they kind of ignore the world around them, and they just become very selfish, and they grow, and they proliferate, and they form tumors, and the tumors are spreading, and you get a disease. If things are going well for you, your immune cells have nipped this problem in the bud. The minute an immune cell recognizes a cancer cell, the alarm bells go off, and that cell gets destroyed. And this is happening all the time, so most of us live most of our lives cancer-free, thanks to our immune system, okay? Now, how does it work at a molecular level? So here's a cartoon that's meant to basically illustrate a very simplified view of that synapse, the point of contact between the immune cell and the cancer cell. And that contact is mediated by molecules. Some of them are on the surface of the immune cell, and so those are the colored shapes, and they're literally connecting with molecules that are on the surface of the cancer cell. They're touching each other. Now, some of the molecules on the immune cell are what we call activating receptors. They're proteins, and they're looking for signatures on the target cell that would indicate that there's been some damage that's taken place. And if you have enough of these activating receptors touching enough of these damage signals, that delivers an activating signal to the immune cell, and the immune cell will respond in what we think of as a positive way, which means it launches a full-blown attack and literally drops a bomb on that cancer cell and destroys it. Now, at the same time, and this was kind of like a bit of a paradox when it was first discovered, at the same time, those immune cells also have inhibitory receptors. These are receptors that are looking for molecules on a target that would be an indicator that it's actually a healthy cell, totally fine cell, not a cancer cell. And if there's enough of these touch points, well, that delivers a signal to the immune cell to stay quiet and go away. Leave the cell alone, everything's fine. And the reason we need to have these inhibitory receptors is because we need to make sure that our immune cells don't make a mistake and start killing healthy cells by accident, because that would lead to an autoimmune disease. That's in fact exactly what autoimmune diseases are. Your immune cells are killing healthy cells, and that shouldn't happen. So these two different classes of receptors are like checks and balances. And it's a tough job that the immune cell has to distinguish good guys from bad guys, and most of the time it works pretty well. So the big breakthrough in oncology that led to immune therapies that are FDA-approved drugs, and some of us might even have benefited from, was recognizing that these inhibitory receptors are often hijacked by really clever cancer cells, right? If you're a cancer cell and you want to live to see another day, you got to figure out, how do you escape from the immune cells? Those cells are patrolling the body. They're looking for you. They're hunting you down. They're trying to kill you. Well, if you can cloak yourself with healthy signals, you might trick an immune cell into thinking you're just fine. It's like the Jedi mind trick, you know? There's nothing to see here. This isn't the cell you're looking for. Move along, right? That's how old I am. So the current immune therapies, which have only been in the practice of oncology for about a decade, they are antibody molecules, they're proteins, that bind either the inhibitory receptor or its ligand, and they block this interaction. So they prevent the cancer cell from delivering the sort of fake healthy news to the immune cell. And by blocking the inhibitory receptors, now the activating pathway can really dominate the situation, and the immune cell can properly kill the cancer cell. And you might wonder, what might be a side effect of an immune therapy that does this? Autoimmune side effects, right? So people who take cancer immune therapies, well, they might have a successful treatment for their cancer, but they might have to live with an autoimmune side effect for the rest of their life. But that is better than dying from cancer for most patients. Now, how revolutionary were these immune therapies? They were pretty transformative, and I'll just illustrate that with an example of some of the clinical trial data that came from Merck and Bristol Myers Squibb. And these are data from around like 2011, 2012, so more than a decade ago now. This was a clinical trial in which patients with metastatic melanoma were being treated with one of two different immune therapies or a combination of the two together. This is what's called a Kaplan-Meier curve, and it's basically a plot of the patients that were surviving as a function of time. Anybody who has heard the phrase metastatic melanoma, and maybe knows somebody, who had to deal with this diagnosis will have fear in their heart. Metastatic melanoma was 100% fatal That was an absolutely lethal diagnosis until immune therapies hit the scene. Because patients that were treated with these drugs, well, some of them died, some of them didn't make it. You can see the survival curve is going down. But at some point, it plateaued. And there were patients in this particular clinical trial treated with these two drugs that just lived and lived and lived. They were cured of metastatic melanoma. And metastatic melanoma and cure are not words that had ever been used in the same sentence before these immune therapies. This was absolutely transformative, and it led to a booming industry in which pretty much every major pharmaceutical company made a drug in this class. And these are now called the checkpoint receptor inhibitors, or checkpoint blockades. They are antibodies that are administered by infusion that block an inhibitory receptor on the T cell, which is one of the immune cells, or its ligand on the cancer cells. And they have absolutely changed the practice of oncology. So much so that when the first few of these drugs were approved by the FDA, you could read about it in all the kind of news magazines, not just the ones that scientists read, like Nature and Science, but even at the checkout stand at the grocery store. You could see from Newsweek that, you know, cancer immune therapy was going to change the course of cancer as we knew it. Here's a newspaper that went so far as to say that these immune therapies might be cancer's penicillin moment. It's a pretty dramatic thing to say, right? When you think about how penicillin changed the outcome of bacterial infectious diseases, and maybe immune therapies would cure cancer with the same vigor, okay? And of course, for those of you in the know, one of our former presidents was the poster child of a patient who got into a phase 3 clinical trial back in 2011. He had metastatic melanoma, Jimmy Carter, in his brain. This was a death sentence, and he was at the time, I think maybe 91. How old is he now? Like 112 or something? I mean, he's still alive, okay? He was saved by these immune therapies, all right? And rightfully, the two scientists who made the foundational discoveries for these inhibitory receptors received the Nobel Prize in Medicine or Physiology a few years ago now. So all of this is sounding pretty amazing, right? But like anything, if you start digging into the actual data of who is cured by these drugs, you start to see that there's still a lot of room for improvement. Because analyzing all the clinical trial data together would tell you that if you're a patient with melanoma, so skin cancer that's metastasized, and you get on one of these protocols for an immune therapy, well, you might survive and you might be cured, but you know, more than half the patients won't be. Okay, so some patients benefit like dramatically, but over half the patients don't benefit. It doesn't work for them. And things are even worse in other types of cancers. So here are some examples of other cancers: renal cancer, so that's you know, like a kidney cancer, lung cancers. Even fewer patients get cured in these other types of cancers. And again, if you're one of the lucky ones, well, that's great, congratulations, right? But most patients don't benefit. And so for the last decade, the big question in the industry is, what are we missing? Why don't all patients get cured by this first generation of immune therapies? Okay? Which brings us back to the cell surface sugars. So as I mentioned, there's this observation dating back to the 1960s that if you look at the complex carbohydrates, the healthy cells have a nice, well-manicured garden of these sialic acids, but the cancers have an overgrowth of sialic acids. Well, guess what? It turns out there is a family of those inhibitory receptors on immune cells that bind sialic acids. And this is a family of proteins that we call the sialic acid binding immunoglobulin-like lectin family. And I apologize because scientists do this all the time. We take a simple concept, make a really complex name out of it, and if that wasn't bad enough, we then give it an acronym that no one knows the meaning of. So we call these the SIG-LEC family. That, I'm sure, is not a household term before tonight, okay? So what are the sialics? These are proteins. You find them anchored to the surface of all the different types of immune cells in your body. In humans, we have 14 members of this family. We give them numbers. So sialic 1, sialic 2, sialic 3, and so on and so forth. They have different shapes and sizes, but at the end of the day, all of them are able to connect with sialic acids. And that interaction occurs in this part that looks like a crescent moon, okay? And this yellow line is the surface of the immune cell, where all the blue stuff is on the outside of the cell, facing the outside world. But then everything below the yellow line is inside the cell, facing the inside world. And this is the stuff that allows the sialic receptors to inhibit the activity of the immune cell. So in my lab, we started looking at these receptors, and in particular, we started looking at the bits and pieces that are inside the cell, and the details are not important. What is important, though, is if you look at the components that are inside the cell, they're almost identical to what's found on one of those T cell checkpoint receptors for which we have FDA-approved drugs. And people in the business would say, oh, this T cell checkpoint receptor and various members of the sialic family have a common signaling machinery. It's the biochemistry that allows them to turn off the immune cell and put it to sleep, like the Jedi mind trick. And the sialics are on every different type of immune cell. And I didn't go through this, but I did say that immune cells are white blood cells. Well, there's different kinds of white blood cells, and it's a bit like the military-industrial complex. So just as, the US military, we've got the Army, the Navy, the Marines, the Air Force, and each different group has its own sort of niche as we fight a battle. The same is true for your immune system. You've got the T cells, the B cells, the macrophages, the neutrophils. These are all the different parts of the armament that we use to fight cancer cells. Every single cell type in this armament has at least one, if not more, of these inhibitory sialic acid binding proteins. So we proposed, and this takes us now back to around the year 2010, that maybe the reason so many cancers are overproducing sialic acids, which are now these trees, is because if they have a lot of sialic acid, they can form really tight connections with immune cells through those sialic receptors. And they could put the immune cell to sleep. And in that regard, this sort of type of biology is analogous to what was already known and where the Nobel Prize was given around the T cell checkpoint receptor, one called PD-1 that binds to a ligand called PD-L1. And so maybe the reason today's immune therapies don't work in most of the patients that take them is because they don't address this interaction. They only address this interaction. So we convinced ourselves that we had found an even better target for cancer immune therapies, which is to somehow interfere with the binding of the sialic receptors to those sialic acid sugars. So we started working on this, all right? And so I'm going to break this down now in a less complicated way, all right? Here's the immune cell. It has sialic receptors, and it also has receptors that are looking for signs of damage. The signs of damage receptors, when they see an indicator of damage, which is what you'd find on a cancer cell, that's a bad taste. And the immune cell wants to kill the cancer cell when it tastes bad. However, imagine that that cancer cell is coated with a thick jungle of sialic acids. Now, a sialic receptor can come to that synapse, and basically, that just tells the immune cell that it tastes good. So it's not so different from a peanut M&M after all, right? It's just the immune cell is tasting the sialic acids, and they taste good to the immune cell. And it's not going to kill the cancer cell if it's receiving all of that good taste. So we have to find a way to make the cancer cell taste bad again, right? So how do we do this? Well, we toyed around with ideas like, okay, let's make a drug let's say it's an antibody molecule, and let's make that drug bind to a sialic receptor and just plug it up. So it's like if the sialic receptor is the mouth and the tongue that wants to taste the immune cell, just close the mouth, stuff a ball in it, right? Now can't taste. The problem is there's 14 members of the sialic family. There's just so many of them. There's so many mouths on so many immune cells tasting the cancer cell. How can you stuff a ball in all of those mouths? It's too much. So instead we said, all right, let's not worry so much about the sialics. There's just too many of them. Let's focus instead on what they're binding to on the cancer cell, which is those sialic acid sugars. How can we strip the sugars off the surface of the cancer cell? And what we did is we made a new kind of medicine that acts literally like a lawnmower. It's a molecule that we park on the surface of the cancer cell, turn on the motor, and just mow those sugars off. Now the cancer cell is going to taste bad again, which gives the immune cell the information it needs to kill that cell. So what's the molecule look like that acts like a lawnmower? It's a protein, and it's a protein that's kind of a hybrid between a monoclonal antibody, which is a very familiar type of drug molecule in the biopharmaceutical industry. But what we did is we attached to that antibody an enzyme called a sialidase. And a sialidase basically is the lawnmower part. It will cut the sialic acid off of whatever it's attached to. It's an enzyme that will do this catalytically. And just to kind of geek out on a little bit of chemistry here, so the type of chemistry that the Nobel Committee recognized me with has a name. It's called bioorthogonal chemistry. And we used that chemistry to basically take the enzyme and the antibody and click them together. Okay? And I won't go through the gory details. But if there's any nerdy chemists in the audience want to talk about this later, we can talk about this. All right. So how does this antibody sialidase fusion molecule behave? Well, we joined forces with a physician scientist named Heinz Laubli at University Hospital in Basel in Switzerland. And he had a very nice mouse breast cancer model running in his lab. And this is a breast cancer model that is known to be resistant to most of the types of drugs that a patient would take in the modern era. And it's resistant to an antibody called trastuzumab, which is an FDA-approved drug. And what we showed is that if we took that antibody trastuzumab, which can attach itself to the cancer cells in this breast cancer model, and we put that sialidase on it so that we can mow the lawn, those tumors are no longer able to grow as vigorously. So this is basically the tumors getting bigger and bigger and bigger over time, regardless of whether you treat the animal with just a salt solution or that antibody by itself. But then if we attach the sialidase to the antibody, those tumors have a really hard time growing. And if you take the little tumors out of the mouse and look closely at them, what you find is the tumors don't have any cancer cells in them anymore. It's just immune cells. All the immune cells have rushed into the tumor and killed the cancer cells. And it's just it's like a little scar tissue. So we were so motivated by this that we decided it was time to try to translate this idea into a human medicine. And to do that, it's often easier to kind of start a company and raise capital at the level that you need to make molecules and put them into human clinical trials. So that company is called Paleon Pharmaceuticals. We hired some amazing scientists at Paleon, and the first order of business for them was to take our prototype molecule, which had 2 enzymes attached to an antibody, and simplify it to make it smaller, which is now 1 enzyme attached to kind of a partial antibody. And also, we had used a bacterial sialidase in our prototype, which is not ideal for a human drug. It's better to use a human protein for a human drug. And so the Paleon scientists have figured out how to take a human sialidase and build it into the molecule. They call this molecule their eagle molecule, because if you squint, you know, it kind of looks like the head and the wing and the claws. I don't know, it kind of looks like an eagle. And here's some data from the company showing that the eagle molecule is also very effective at keeping these tumors from growing out of control. The blue curves are mice treated with the eagle molecule. The black curves are mice that were treated with just kind of other controls. And you can see the tumors grow very quickly with the controls, but when the eagle is there, they slow down. Sometimes the mice will relapse. Sometimes the mice are cured. But in this graph, what we did is we combined the eagle drug with the conventional immune therapy targeting the T-cell checkpoint receptor. And when we put the two together, now we can cure every single mouse. So I'm excited to say that Paleon Pharmaceuticals has a green light to put their first eagle in a human phase 1 clinical trial starting in Q2 of 2025. So wouldn't it be cool if you invite me back here for your 30th anniversary and maybe we have a medicine? You know, that would be great. So with that, here's like the take-home message, because I do teach and this is a lecture, so here's what you should remember. This will be on the quiz. First of all, hopefully now you know that all your cells are coated in sugars. That's, I think, memorable, right? Like the peanut M&M. I've also taught you that, you know, cancers have altered sugars. In particular, they have jungles of sialic acids that helps them survive by putting the immune cells to sleep. And we know how that works at the molecular level. Those sialic acids are binding to that family of receptors called the sigleks, which are on immune cells. And it's through the sigleks that they deliver the Jedi mind trick that hypnotizes the immune cell into leaving them alone. And understanding all of that, we had an idea for a new kind of immune therapy where we make lawnmower molecules that mow the sialic acids off of the cancer cell so they taste bad again, and we deprive them of their hypnotic powers. And hopefully we'll make a medicine from that. So with that, I'd like to thank, this is my group dating back many years now. That's a fairly recent group photo in a building that we designed, very much inspired by the Biodesign Institute. Open lab space, lots of windows and open views. This is a group meeting during COVID, lest we forget what it was like to meet outside 6 feet apart from each other with masks. And then after the Nobel Prize announcement, a bunch of my alumni connected with each other and organized a Zoom party on very short notice. We had 150 people on Zoom from every time zone on the planet drinking a toast of champagne. And I owe a great debt of gratitude to all of these wonderful coworkers who did the work that I had the privilege of talking about tonight. And with that, hopefully I've timed this so that we can have dessert after we learned about sugars. Thank you.
Questions? So we have time for a few questions. Does anybody want to ask a question? I'll start. Oh, that's one.
Oh, there's a mic.
Thanks. I'm just curious, do you see an increase in the autoimmune side effects with the combination of the two?
That's a great question.
Okay, I don't want to.
It's hard to know from animal studies because the immune systems, especially of mice, are so different from humans that you tend not to be able to get a read on those kinds of side effects. We know what to look out for, certainly. And I think autoimmune side effects should be expected. I think we should anticipate some of that. And really the question is, how severe will they be? Now, with the current therapies, the T-cell checkpoint inhibitor therapies, as they're called, common side effects are, if you're treating melanoma, often vitiligo is a side effect. That's where people lose their pigment, which is, you know, cosmetically disturbing, but physically harmless. So that's not so bad. But sometimes patients will end up with type 1 diabetes, which, you know, is a chronic condition. They'll have to manage that for the rest of their life, but they have a life, right, in which to manage that. Some of them end up with ulcerative colitis, things like Crohn's disease. So these are the things that you'd have to watch for. Unfortunately, some of these autoimmune side effects unfold on a fairly long time scale. So you might not even see them in the sort of shortened course of a clinical trial, but then they emerge over a couple of years, right? So it's kind of a longer term proposition, but we'll have to be very watchful.
Thank you.
Oh, we have one over here too.
I'm coming, I'm right here. Well, first, congratulations on the clarity of your delivery. It's absolutely amazing for non-scientists. I mean, really well done.
Thank you.
A quick question, assuming that conjugate works in the early human trials, the difficulty of scaling up a rather complicated molecule?
That's a good question. So the antibody enzyme fusion is a complicated molecule. It's not a conventional antibody. The team at Palion spent the better part of 10 years. We formed that company in 2014, so it's been 10 years now. And the better part of that time, they have spent engineering that protein to basically make it developable as a human drug. They had to do a lot of sort of mutagenesis on the enzyme to make it stable in the plasma so that the protein would express at high levels in the manufacturing context. I think the last count was they made 350 plus different constructs to finally arrive at their development candidate. That's what it took. So yes, it was complicated. But they now have a procedure, and they have a molecule which they express at something like 7 grams per liter, which the experts will know that's pretty good. And it has a plasma residence time in non-human primates that's on the order of about a week, which is not as long as a conventional antibody, but probably long enough to do once every other week dosing.
I think there's one.
Thank you for that wonderful lecture.
Yeah, you're on.
Okay. I feel like I learned a lot. I really appreciate that. I know there's a lot of complexity here that you had to sort of gloss over, but one of the ones I'm wondering about is that cancer is notoriously heterogeneous. From cancer to cancer, how often is this sialic acid the mechanism of immune evasion? And within a cancer, how much variation is there between cancer cells for using this? And in particular, is there evidence that they would switch to CTLA-4 as an immune evasion if they PD-1 and sialic acid?
Yeah, these are great questions. In terms of heterogeneity, first of all, some cancer types have a higher prevalence of the hypersialic acid phenotype than others. So for example, and we know this because we've done a lot of immunohistochemistry, looking at sialic acid expression levels in tissue microarrays and tumor microarrays from biopsy samples. So melanomas, like 90% of melanomas have very high levels of sialic acids. Breast cancers, ovarian, pancreatic, colon, bladder. And then there are some tumor types where it's less prominent. For example, head and neck cancers, renal cancers. You tend to have, maybe 50% or 40% of the patients have this phenotype. Then within an actual tumor, it's interesting to compare what the immunohistochemistry looks like, the tissue slice looks like, when you stain it for sialic acid versus, for example, PD-L1. A PD-L1 positive tumor that's a patient that's eligible for Keytruda or Opdivo or one of these drugs. Positivity for PD-L1, if you actually look at the tissue, it's like little blobs of PD-L1. It's not like coverage. If you've seen these, you know what I'm talking about, right? It's very patchy. By contrast, the sialic acids are a blanket. They really cover the entire tumor. So it's much greater coverage. Now, whether that translates into, a different potency of a signal or something like that, we don't know, okay? But just qualitatively, this is what you see. And so when we run our clinical trials at Palion, we will be analyzing patient biopsy samples and selecting patients based on the intensity and the coverage of their tumor tissue with sialic acids. So we try and stack the deck to people that will respond. If a patient doesn't respond to our therapy, or if they respond but then relapse, could it be because those cells have evolved a different mechanism of immune suppression, CTLA-4, for example, or something else? Probably. I think, tumors usually relapse. With immune therapies, you might get a cure, but patients also have relapses. And the truth is, it's evolution, right? A cancer cell will evolve to find a way to survive if it can. And that usually means finding other ways to suppress the immune system. So I have no illusion that lawnmowers are going to cure all cancers now and forever. But I think we can maybe get from 50% response rates, hopefully more like 75 or 80% response rates. That I would think of as a win.
Good. We got some over there.
So I'll ask a geeky chemistry question.
There's a mic right behind you. There you go.
I'll ask a geeky chemistry question since you gave us the bait. Did you learn about the Pictet-Spengler reaction from Rappaport?
Oh, that's an extremely geeky question to ask. So I think I might have, so the Pictet-Spengler reaction, what is it? Organic chemists like me historically have tendencies of naming reactions after ourselves. And that was certainly the case 100 years ago. Naming a reaction was a big deal. And then in my generation, the challenge was to learn and memorize all those named reactions based on their names. So when I was a grad student, I learned 100 or so named reactions, including the Pictet-Spengler reaction, which was first published in like 1905. That's how old that chemistry is. Henry Rappaport did teach my first-year organic synthesis class for first-year grad students. He might have been the person who taught me that chemistry. He certainly had a passion for indoles and amines and amino acids and that kind of chemistry. And what I love about the kind of chemistry that has proven useful in biology in our lab is a lot of it is based on chemistries that were more than 100 years old. And the reason I love that is because it's a good reminder that when Pictet and Spengler first published on doing indole alkylations, they never in their lifetime would have thought, oh, and someday somebody will stitch an enzyme and an antibody together using this chemistry and make an immune therapy for cancer, right? That was nowhere on their grid. They were basic scientists doing basic science, curiosity-driven basic science, inventing reactions to build molecules in new and different ways. And like most of us, they wouldn't have known the impact of their work in their lifetime. Just like, I'd like to think I know the impact of my work, but I don't. None of us actually will know the true impact of our work because a lot of it will happen well after we're gone from this earth. So it's a good reminder that we need to invest in curiosity-driven basic science, even if we don't know what the impact will be yet. That's what I think.
So that's a great talk. I learned a lot about sugars. I like them just like anybody. But on a genetics basis, is there a predisposition to respond to these therapies that we know about where certain families could do much better than others? And could that be the case in your clinical trials that you look for that to make it more personalized?
So that, the holy grail is to be able to predict which patients will benefit from which therapies and choose the right patient for the right therapy. With the immune therapies, we're not there. The fact that half the patients we give them to don't respond, and we can't predict who will and who won't. And meanwhile, the non-responders, their cancer is still there, plus now they have autoimmune disease, right? So, we don't want to just give medicines to people who are not going to benefit from them. So we're not there yet. I mean, there are some indicators that, so you remember the cartoon, there's activating receptors and inhibitory receptors. If you block the inhibitory receptors, nothing will happen without an activating signal, right? So you have to have an activating signal and block the inhibitory signal. And I think some patients who don't respond to this first generation of therapies, they might not have a strong enough activating signal. So there's a lot of interest in drugs that go after the activating receptors and agonize them. And maybe you'd want to pair that with a blocker of an inhibitory receptor. But we're really not at the level where we can make good choices about the right patient for the right therapy. That's very much the leading edge right now.
We've got one over there.
So you started your presentation off about blood types. Is there certain blood types that are more susceptible to certain types of cancer or immune therapy that works better on certain blood types?
This is a great question. I don't know of any relationship between blood type and cancer, but there definitely are relationships between blood type and susceptibility to certain infectious diseases. And in fact, the theory of blood types goes, so whenever you see like stable variations in the human population, like stable genetic variations, usually that means that there was some evolutionary pressure from pathogens. That's usually how that works. Like sickle cell disease. That's probably the most famous example. Why do so many people on this planet carry like one allele of the sickle cell mutation? Shouldn't that have just been like filtered out of the human population, given that sickle cell disease is so deleterious? And the reason it persists is because it's actually an advantage under pressure from malaria. So a patient who carries one allele of the sickle cell mutation is more resistant to malaria. So that's how you, by natural selection, create a new gene pool and propagate it. Cystic fibrosis, same thing. If you carry one mutation in your cystic fibrosis gene, you actually have some resistance to various diarrheal diseases, foodborne types of pathogens. And that's why that persists. So blood groups, why do we have A and B and AB in big human populations? People think it's because when pathogens enter our body, it's helpful if they can mask themselves to look like us, right? That's how they escape our immune system. And since sugars are the coating of cells, the first thing your immune system sees are sugars. So if a pathogen can figure out how to decorate itself with the same kinds of sugars that your cells have, it can hide and it can thrive in your body. So there's some theories that there was bacteria that were able to create a sugar coating that looked like a blood group, right? We don't know which one, but let's just hypothetically say suddenly a bacterium knows how to make the blood group A carbohydrate. And that bacterium can now infect you and escape your immune system because it just looks like a blood cell. It's got blood type A sugars on it. Until a human spontaneously has a mutation, so now instead of making blood type A, they're making blood type B. Now their immune system is poised to recognize blood type A as foreign, and they'll kill that same bacterium. So there's a tennis match between us and the pathogens. And sometimes we score a point, and sometimes they score a point. And this goes back and forth over millions of years, and that's how you get the blood groups, right? So it shouldn't surprise anybody that certain bacterial infectious diseases are more successful in a person with this blood type versus that blood type. It seems to be the case for the bacterium that causes gastric ulcers and also gastric cancer, which is Helicobacter pylori. And during the height of COVID, a couple of papers came out showing that people with certain blood types had a higher propensity of getting severe COVID versus mild COVID. So I think that's where you would look, is in infectious disease, maybe not necessarily in cancer.
Hi. Yeah. By the way, thank you so much for giving such a good analogies. So I really like the cartoons that you showed. But a question that I have is that has there been any studies in terms of depriving the cancer cells of sugar molecules? And I believe you called it sialic acid, if I'm not mistaken. And mainly, my question out of curiosity is that has there been any studies where patient cancers were studied, for example, finding a correlation between patient cancers and the nutrition that they are taking? For example, if they're taking less sugary nutrition, that will improve their condition, because you're actually going to essentially deprive the cancer cells from the sugars that they're hiding. Basically, they're hiding themselves from the from immune system. Just mainly, I just put it out there. Was wondering about that.
It's a really good question. It kind of begs the bigger question. At the start of my talk, I said, when you think of sugars, you think of food, but we're not going to talk about food. We're talking about something totally different and unrelated. We don't actually know if there's truly no relationship between your sugar intake and what the forest on your cell surface looks like. There could be sort of an indirect relationship there. I will say that there have been a number of studies showing that certain cancers, in fact, many cancers, are really fueled by sugar in their growth, and they're fueled by glucose, which is that's like the sugar from which we derive energy, is glucose. In fact, some cancers are what we call addicted to sugar, and it's called the Warburg effect, right? It's cancer cells often shift into a sort of metabolic state where they just need sugar, more and more sugar. They're like totally addicted to sugar. They don't want to eat fat, they don't want to eat protein, they just want sugar. And if you can put a person on a very low sugar diet, like a keto diet, then in principle, maybe you could starve out these cancers and they wouldn't grow as quickly. But I don't know whether those types of experiments have been done on the scale of human clinical studies where you could actually get a statistical read on that. But in animal studies, yeah, there's some stories like that. I mean, you probably shouldn't eat too much sugar anyways. Sorry, that maybe not the right time to give this advice. Yeah.
So again, thank you. This was just an amazing lecture. You talked about how these treatments can trigger autoimmune, and so often we say we don't know what causes autoimmune.
Yeah.
So is there a possibility that the research that you're doing now will move beyond cancer and help us address or prevent triggering autoimmune signals so that we can deal with the massive chronic disease problem we have?
Yeah, that would be great. You're right. Autoimmune disease is a huge burden on the health system. And autoimmune diseases include everything from rheumatoid arthritis, Crohn's disease, lupus, psoriasis, multiple sclerosis. I mean, there's so many different autoimmune diseases. Put them all together, it is a huge burden. And yeah, I think people have learned a lot about autoimmunity by studying cancer immune therapies, and not just ours. I mean, the PD-1, the first generation therapeutics have taught people a lot about autoimmunity. The sialic receptors, there's some variations in the human population, and some of the variants of the sialic receptors have been associated with higher rates of ulcerative colitis and Crohn's disease, right? So we might actually already have hints from human genetics that the sialic receptors are important in protecting us from autoimmunity. And if they are more or less functional, you might have a higher risk. I'll tell you where the sialic receptors are very important in normal, healthy biology is in pregnancy. So pregnancy is a situation where a foreign tissue is growing in a woman's body, and her immune system has to be taught not to reject it, right? And the sialic receptors and their ligands are very populous in the placenta. And they're basically making sure that the immune cells, any immune cell that gets into the placenta is immediately quieted down, like shut it off, turn it off, inactivate it, hypnotize it to mitigate the risk of fetal rejection. That's the normal biology. I mean, it's not like, we don't evolve immune systems so that cancer cells can thwart them and make us sick. That's not the purpose of these receptors. The purpose is to fight pathogens over here and protect us from rejecting a pregnancy. Because, for the human species to thrive, all that really matters is pathogens and pregnancy. Whether you get, rheumatoid arthritis when you're 65 is not relevant to the survival of the human species. But surviving bacterial and viral infectious disease and then be able to have a pregnancy, those are two absolutely vital things. And that's, I think, most of the evolutionary pressure that's created the system as we know it came from those two things.